Hi, my name is Tanner Bland. I'll be covering EPAS1. EPAS1 is an HLH transcription factor that regulates oxygen levels in low oxygen environments. EPAS1 creates HIF2 alpha protein, which is about 870 amino acids, and it's roughly 100,000 base pairs in gene length. EPAS1 is found in most complex metazoan taxa, including fish, birds, and mammals. And it's really important for understanding low oxygen adaptation. EPAS1 is regulated by proteasomal degradation, where subunits of HIF, the larger protein that HIF2 alpha creates, are constitutively expressed but are degraded if in high enough concentration. And so we have this gradient of expression where in high oxygen environments we have low concentrations of HIF2 alpha and vice versa. This increases or decreases the amount of HIF in the body, which in turn increases or decreases the amount of oxygen that's being transported around the body. EPAS1 is involved in erythrocyte formation, in blood vessel formation, and in hemoglobin concentration, as well as embryogenesis. EPAS1 is located on chromosome 2, location 21, and is 97,000 base pairs. It's inherited as an autosomal dominant gene for most. There's a few cases of EPAS1 not being inherited as a dominant gene and being absent. However, this is very rare and usually leads to a lethal phenotype within just a few years of life. These people usually have a very low quality of life. EPAS1 interacts with many different developmental genes, namely ARNT, which is involved in developing tissues, organs, and in cephalization. And by regulating the amount of oxygen that goes to different areas of the embryo, EPAS1 can actually cause cephalization by concentrating oxygen in certain areas, and it acts as an activator for ARNT by increasing or decreasing oxygen levels. And ARNT is sort of switched on or off by the determining oxygen levels within the cell, which in turn activates or deactivates EPAS1. So lastly, we're going to go over a quick summary of my paper. And so a major finding of all of these papers just across the board was that there are variants in Tibet and Bhutan that have a high resistance to hypoxia in their EPAS1 gene. This creates a phenotype that has high blood cell count, that has high blood vessel count, and has high concentration of hemoglobin in their bloodstream. These phenotypes actually are called super athletes. They are typical among people that have super athlete-like capabilities. They are able to transport oxygen in their body much quicker than most people, and that gives them sort of an advantage doing athletic activities. These also granted, EPAS1 also granted some major insights by studying these variants. In human ancestry, as we can see on the right in the figure, Denisovan haplotypes are present in some of these Bhutan and Tibetan populations. The black in the pie chart represents an relatively unchanged Denisovan haplotype in comparison to the colored phenotype that we see in the rest of the pie charts. We can see that as we increase in altitude, we actually see an increase in the presence of the Denisovan haplotype. And this actually gave insight into how early humans may have interbred with ancient human species like Denisovans in the past to be able to cope with high altitude environments. EPAS1 is also a great candidate for gene therapy in COPD. COPD is characterized by low oxygen delivery efficiency, and upregulation of EPAS1 can help cure that efficiency degradation in COPD patients by increasing their blood cell count. And in colorectal cancers especially, these cancers thrive in hypoxic environments, as one study in my paper found. And by increasing EPAS1 within these cells, you actually find that EPAS1 serves as almost like a tumor suppressor gene. In fact, in many colorectal cells, tumor cells, you can find that there is methylated EPAS1 genes on their DNA. And so this sort of proves that EPAS1 plays a major tumor suppressor role in colorectal cells, because tumor suppressors are thriving in methylated EPAS1 environments. And finally, the most interesting area to me was that this lends major insights in the agricultural sector. HAPH, which is high altitude pulmonary hypertension, found in many high altitude bovine populations, is less common in cattle that are expressing an EPAS1 variant that's very similar to humans. And people are sort of using these variant phenotypes to treat HAPH in lower altitude cattle. And this isn't Cape buffalo or water buffalo in Asia. This is like Rocky Mountain cattle that were grazing in Montana and Colorado. So some really interesting agricultural research that's really close to home. And finally, some of this data lends insight into convergent evolution events. Ducks and cattle across the world have actually developed the same sort of phenotypes based on their proximity to humans. And so this sort of gives insight into how agriculture and the domestication of livestock over time with humans has caused cattle and ducks and similar livestock to be able to adapt to high altitude environments. The study that I looked at was using the Andes as a model, but this was extremely applicable to pretty much any geographic area that was high in altitude, like Tibet and China. Well, thank you, and I hope you enjoyed.